All right. Welcome, everybody, to another Journal Club session presented to you by Life Extension Advocacy Foundation, lifespan.io, broadcasting from New York City at the Kambar Center for Biomedical Engineering at the Cooper Union. I'm Dr. Oliver Medvedik. Joining me is Steve Hill and Fatima Hill in the background. And I don't think we have anybody else joining us. We're doing it at a slightly different date because we have a lot of stuff on our plate, and we're going to be going to a conference in Berlin next week, an Undoing Aging conference. So we're going to have lots to report, no doubt, when we get back from that. But what we're going to report on today is yet another awesome paper. And let me just pull this paper up here, share my screen, adjust screens. There we go. This is a paper that just came out fairly recently. Young bone marrow transplantation preserves learning and memory in old mice. Das et al., number of authors. This is from Cedars-Sinai Medical Center over in sunny California. And basically what this paper is looking at is trying to kind of narrow down what happens when we do parabiosis. So specifically heterochronic parabiosis, as it's called, meaning taking differently aged organisms, animals, fusing their vasculature together and basically having the blood supplies intermingle. And swapping, young blood for old blood and, has been done for quite a long time, actually many decades. It's been observed. And there was a clinic, I think, that was offering, plasma transfusions from young donors to old donors. This is humans. There's also a clinical trial that just ended recently that's referenced. I'll get to that in a moment, involving Alzheimer's patients. I'll just get to the punchline. It was a very short trial and very few people. So they weren't really looking for efficacy. They were looking for safety and kind of compliance. And that seemed to go well. So we'll no doubt be seeing more of that in the near future. Bottom line is there are things in young plasma that seem to benefit, older individuals, older recipients. And what this paper set out to do is try to kind of zoom in on what some of these constituents might be. And one hypothesis was that what they're doing is not doing a parabiosis study, but actually transplanting young bone marrow into old mice. And as the title, tells you right off the bat, it's preserving learning and memory in old mice. And what this paper sets out to do is try to figure out, well, what in young bone marrow is precisely the causative agent. And again, I'll kind of skip to the punchline. It turns out that it's reduced levels, or at least one of the things that's implicated here is reduced levels of a chemokine, a ligand, CCL11, not beta-2 microglobulin, which they haven't seen reduced levels, I think, in this paper, but rather CCL11. And what CCL11 is, a chemokine is basically a molecule, and in this case, a small protein, and it binds to a receptor, and the receptor is from the family of G protein coupled receptors. Just stop sharing here. That's a G protein coupled receptor. It's found in eosinophils, I think among other cells. These are white blood cells, and they're involved in allergic responses and I believe inflammation as well. So it makes sense. You have a lower ligand for this receptor that promotes inflammation by activating microglial cells, basically these phagocytic cells that may promote inflammation. So you lower this subclass of cells, and you have beneficial effects. Okay, so let's take a look at the data here and see how this holds up. So what they do is they first kind of do these transplantations, and then they jump right into the, I kind of like this because a lot of papers, they go kind of backwards. They kind of build, and then you see, well, it didn't really have that much of an effect. Here they basically, they did the bone marrow transplantations, and they went right into looking at cognitive function, right, and seeing, is it doing anything? And then if it is doing something, let's see if we can kind of narrow down what exactly is happening. So let's share the screen here. Okay. So we're just going to jump right into figure 1. Okay, so here we have kind of the layout of their experiment, figure 1A. What they do here, so the way they ablate the old bone marrow is they irradiate these mice with gamma radiation, and that's typically done in human patients when they do bone marrow transplants. And it turns out that the hematopoietic stem cells, which are the stem cells that basically lead to the differentiation of all your blood cells, are very sensitive to gamma radiation. So you'll pretty much wipe them out. And what they do is they mention in the paper that when they gamma irradiated these mice, they didn't add any kind of head shield. So what they're going to be looking at is cognitive function, and the reason this is important is they want to mention that when they do this, it's when they look later in the paper, it's not going to be so the treatment that they do is not going to be I'm trying to write a sentence here in this paper, conducive to neurogenesis, right? So let me find the exact one in this paper. So I got it underlined here, and it's scattered. Well, I'll get back to this. You can find it, Steve. But anyway, so what they do is here, after they do this radiation, they do transplantation, and they basically hematopoietic reconstitution. So take old bone marrow and put it into, young bone marrow, sorry, and put it into into old mice, and vice versa. So they have young and old are your controls, basically, no transplantation, and then old plus bone marrow transplantation, and old plus young bone marrow transplantation. And so they give this animal some time before they do behavioral testing. Now, 18 months is an old mouse, so after 6 months, these are kind of really old mice. I guess 24 months, 2 years old, and then they after this transplantation, they do all the assays. They sacrifice the mice, but so here they basically start out with activity assays, and kind of in line with what's been seen before. So all these squiggles are basically, I guess, tracking of a mouse running around in a cage. And I think I'm not sure, they tell you how many mice there are here. You can dig down here, like n equals 32 to 51 mice. The numbers kind of vary. So later in the experiments, they have less mice that they work with. But you can see just by, if this is representative, you can see these two look vastly different from these two, right? So old mice with young bone marrow look like young mice, kind of. I mean, it's definitely different from this. So they're more mobile. They have these activity assays here, which here are these little stars that tell you what the p-values are. So the significance. So I'm just gonna, can't see this because my face is hiding this, but p-value of 0.001, so pretty significant. So activity beam breaks. So I guess this is a mouse standing up, distance that the mice traveled, mean speed, how fast they went. So you could see that from the gray on this side, which is your young control, and the red on this side, which is old plus young bone marrow transplantation, you could see just by glancing at this that you have this significant, this non-significant difference, meaning that it is performing, more like a young mouse rather than these controls here, which is old plus old BMT and old control. So, that's a very good start. So right now the mice are behaving, at least when it comes to motility and functionality, like young mice. But of course, this paper is really more concerned with cognition. So this is when we get into figure 2, where they look at, they do a variety of cognitive tests. And again, these are, physiologic tests. What we got here? Chat. Same has been seen in senolytic studies. So I'm looking at the chat here while I'm reading this. Same has been seen in senolytic studies where old mice hug the edge of the cage and younger ones explore more. Yes. So I think what we'll see more and more when these, it's good to see all of this kind of consolidation happen, right? With all these loose threads coming together, and we can talk more about what good follow-up experiments there are, but certainly inflammation is a key factor here, and this is a big implication, at least with regards to when they look at this CCL11 chemokine ligand later. So here they have a variety of cognition tests, and kind of what they do is, so alternation. So, I haven't done any of these tests with mice, so I'm just basically basing this on how it's described in the paper. You have like a Y-shaped, like sort of basically a fork in the road, right? And the mouse gets to the fork in the road, and the mouse is like, which way do I go, left or right? And it's trying to decide. And I guess, a younger mouse is more curious, so it alternates between the two. It's like, do I go left? Do I go right? Do I go left? Do I go right? So it's percentage alternation. If I'm reading this right, maybe I'm reading it backwards. But just by glancing at this, you could see that the old with young BMT, it's, again, you got big error bars here. So, so this, again, this tells you when you do these types of tests that even though these mice are all, I'm assuming, one strain of mice, you need a pretty large sample size, to see these kind of differences. But again, in this Y maze or study, you have a similarity between old, young and old plus young BMT. What they do in B and C is basically so now they're going from kind of decision making to memory, and this is where you seem to have, I guess, a lot of neurological defects in the hippocampus, which is involved in memory. So they're going to kind of go into that at the cellular level in the next figures, but right now it's based on behavior. And basically what they do is they give the mice an electroshock, right? So they zap the mice and, a bit of a pain response. And the mice are put into a cage, and so there's context and cues. So either the cage, I don't know how the cage looks similar. I guess they put like little furniture or something in there that the mouse can recognize. Oh, this is the cage where I get zapped all the time. And then there's a cue. So prior to the zapping, they play a little tone, right? The tone tells the mouse, hey, you're about to be zapped. So the time frozen is when the mouse gets into a cage where it's, not comfortable because it associates it with a pain response. It's going to freeze. It's going to be like, whoa, I'm someplace bad. This is triggering, post-traumatic stress syndrome for a mouse, and the same thing with a cue. So the bottom line is that with time frozen, it's a better memory of the event, right? So the more the mouse is frozen after this entrainment, then that means it's remembering. And the young mice, you could see the spread here, and then old and old BMT, and then you could see this frozen response is this improvement in memory, or a recognition of a bad event. The cue, I don't know if that's not at, I don't know if they're able to associate the cue as well. I have to take a look. I'm not sure if there's a significance in this cue response. They do another cognition test where they, I think it's a maze, and then there's like there's like an escape hole. I don't know if this is like a water maze, but basically the number of errors goes down. So this is entrainment, right? So these are mice that are, day 1, day 2, day 3, they're trying to train these mice. And I guess most of these, I don't know why these error bars are so big. Most of these mice are, well, these are old. They're learning over a period of time, the errors start to drop. What they do next is there's a pause, and then they basically put the mice back in and see if the mice could remember, where that escape hole was. And you could see that after that delay and putting, the mice back, the number of errors is pretty low for, a young mouse, but, there's a big spread for old and old plus old BMT, and not as much for old plus young BMT, right? So they're remembering. And then they have something kind of similar, let's see, day 8, day 9. This is a little different. It's basically they reposition the hole, right? So they kind of mess with the mouse. They bring it back day 8, so the mice have this many errors, and then they kind of reposition the hole, and the mouse has to now refine the hole again because it's now in a different location. After kind of getting used to looking for it in, it's similar in some location. And you can see that when they do that, the mice that are young, they, you know, its number of errors is much less. At least you have a statistically significant difference, even though there's a lot of overlap here, with old and old plus old BMT versus old plus young BMT, which, you know, again, so that's pretty good, right? So something is happening with, you know, they're not transplanting the plasma or fusing the vasculatures. What they're doing is they're doing a bone marrow transplantation from one mouse to another. So that's rather unique here. And they want to, you know, so you're reconstituting, you know, it's sort of like a parabiosis, but sort of parabiosis intrinsic, because you're essentially reconstituting a young bone marrow in situ in this mouse, right? So you get the young plasma and everything from the get-go. Well, sort of, right? Because the bone marrow is still dividing in the context of an older mouse. So not quite. All right, so let's go back to sharing the screen. And now they're like, okay, well, we're getting some effects, so let's see what's happening inside of the cells, right? So synapses. So getting to the punchline, they're not seeing more neurogenesis, so not seeing more, so they're doing staining here. So these are new, gotta get my crib sheet here. But we have neuronal numbers in staining. So they're doing a stain here in CA1 and CA3. These are regions, I believe, in the hippocampus. I could be wrong. And they're looking at, you know, all the different mice here. So young at this end, young, old plus young bone marrow transplant, and, you know, old and then old bone marrow transplant. And the number of neurons, you know, the number of neurons isn't changed. They're saying here, I don't know, they're looking at another hippocampal region. They're saying that these are not significant. This is young, this is old. I'm not sure why there's a slight shift up. They kind of mention it briefly in the paper. I'm not sure if there's a significance to having slightly more neurons, even though it says non-significant. There looks like there's some kind of a trend here. And then they look at DCX cells, and this is a stain. So they look at, they do also in the paper BRDU staining. So BRDU is a stain for dividing cells, and DCX is for, I think, new neurons that are basically, you know, growing. So these are young mice, and then when you have old mice, you don't have any more neuronal growth happening. So the neuronal growth is kind of ceased. So there's no new. So basically, the bottom line is there's no new neuronal growth happening in these hippocampi. But what they do notice is, I guess, better synapses, more synapses, or the reestablishment of synapses. So this is V-GLUT plus Homer1. These are weird terms, but basically this one is for presynaptic, one is for postsynaptic connections. So these are, I think these are, trying to jog my memory here, trying to jog my synapses here, but these might refer to, you know, neurotransmitter channels, or ion channels. And one is on presynaptic, one is on postsynaptic, and this is, you know, percentage compared to young. And you could see that when you have young here, and then this is your old plus young, you know, bone marrow transplant, you have a slight increase, and these fuzzy images here are supposed to be dendrites. And these boxes are, you know, they correspond to the different bone marrow transplanted, you know, mice. And what you are seeing here are things called dendritic spines. And basically they look like little branches with little broken off branches. And those little nubs are where I believe synapses tend to form, right? So you've got all these connections. So kind of the more branchy or shrubby looking this thing is, the more synapses there are. So you can see that, you know, here is supposedly a representative sample from old, and then old plus old. And this is young, and then old plus young. So when they're looking at these spines, this is, so this is a stain for a number of synapses, and this is actually the spines themselves that are indicative, I believe, of synapses. All these stars are, p-values that they've done over bunches of different neurons that they've counted, and spines per micron. So, it goes up, not as high as young, but, these error bars are much smaller, and it's definitely significantly higher. So you seem to be preserving neuronal, what's referred to as neuronal plasticity, right? Meaning that the neurons are able to kind of remake connections. And why are these connections broken? Well, they look at messenger RNA expression levels of these factors here, these complement factors, C1qa and C3. And what these are indicative of, or these are markers that, these are cells that are being basically targeted, I believe, for, I want to say phagocytosis, but I want to check this here. So I think it's phagocytosis by glial cells, or they're being attacked by the glial cells. My papers are all scattered here, but I'm going to leave it at that. I'm going to double check that in the paper. But you can see here that the markers for essentially targeting for glial cells are much lower. Messenger RNA levels in the neurons are lower, and in the neurons that from mice that have done the bone marrow transplantation from young mice, the old mice, these C1qa and C3, and again, and this is a correlation with synapse. So Homer 1, I believe, is postsynaptic neurons plus C3. And you can see that, in these neurons that are specifically, not just all neurons, but specifically, one's postsynaptic, the numbers of this complement protein go down, right? So, okay. So it looks like they're being protected. So they're in this figure here, figure 4, they're moving on from, what's happening to the neurons to what's happening with cells that are actually implicated in kind of promoting this damage, right, to neurons and synapses. And that's these microglia, right? These are the supportive cells that are, involved in phagocytosis and potentially inflammation. And basically, as the first sentence states in figure 4, young bone marrow transplantation reduced aging-associated activation of microglia. So what they do is now they basically want to look at microglial cells and show you what's happening to them and show how this indicates that they are less kind of aggressive after the bone marrow transplantation. So I'm going to stop sharing here. And let's see, we have a chat here, something. Luca Biasco, hello everyone. I see 2 points that should be raised. I don't know if they're addressed later in the paper. 1, if I got figure right, the cognitive behavior is measured at 3 to 6 months after transplant. I believe it's 6 months. This would be a relatively early time point after transplant, where stressed hematopoiesis is still in play as a result of the big push towards fast food. They check for differences in cognitive behavior at later time points where steady state hematopoiesis is in place. I'm not sure. I think at that point the mice are dead, but they're checking them at 24 months. 24 months is pretty old for a mouse. So they're doing it 6 months. So, maybe they could have done this at an earlier time point, right? Because they did it at when the mice were, what, 18 months old? So, that's all I can say off the top of my head. They could have, you could use longer lived organisms. So that's a good follow-up. You could probably use rats or a longer lived strain, but I'm not sure. Let's see what, let me go to this paper, and I'm just scrolling here. Well, let's see, what mice did they use? Male wild type what? CD45.1 and CD45.2 congenic strains. So let me Google that for you. How old does a CD45 congenic strain live for? Strain age. It'd be funny if it lived shorter than the lifespan of the mice in this paper. Yeah, I don't know. Got some experiments where they're they're 22 months old. Certainly not dead at 22 months, but yeah, I don't know. If somebody can look that up and let me know. Then yeah. Let's go back to, so figure 4, young DMT reduced the aging. So now they're looking at glial cells. Steve is typing something. Let's take a look here. All right. Oops, wrong one. This one. Scroll, scroll, scroll. Close your eyes. I'm skipping over stuff. Okay. So figure 4, they're looking at glial cells. So they're looking at expression of, so astrogliosis. So these glial cells have a marker called GFAP. Trying to look up the acronym here. That's the one thing about molecular biology. There's tons of acronyms. Some acidic protein that's associated with glial cells. And the more of it, the more active they are. So I'm going to leave it at that. So the number of glial cells they looked at in various fields of view are the same. So if I'm interpreting this right, even though there's the same amount of glial cells, they're expressing more messenger RNA for this factor that indicates activation of glial cells in old mice and old mice with old marrow, but not in young mice, or young mice here in the gray and the red, which is young old mice with young bone marrow. More on the chat. They are variants of the C57 black 6, aka black 6 mice. Great. How long does a C56 black 6 mouse live?
You'd think I'd know that, seeing as we experimented with them. I'll Google that for you. But black 6 are very standard mice. They use them a lot. I think it was about 80+% of Jackson Lab sales are this particular type of mouse. And the great thing about them is they're not particularly special at all. There's nothing really special. The only unusual characteristic is they are one of the few mouse strains that actually drinks alcohol willingly.
Oh, really?
Yep. There's dating back to the early part of last century, there's a little anecdotal report from a researcher who describes them going on the rampage and somehow managing to get very drunk. But yeah, they do actually drink alcohol. Which is quite fun, but they're these are a particular strain of or substrain of these. They're a congenic strain, CD45.1, 45.2, but that's the base strain. So they're not progeric.
Okay,
which is good news, because obviously a criticism with progeria mice is it's not real aging, which is fair enough, isn't it? Really.
Okay.
But they're very commonly used in aging research.
All right. Okay, so back to the glial cells. So the one thing that they mentioned about so when glial cells are active, when they're actually doing things, there's a number of different kind of I guess let's call it parameters that are measurable, right? And one thing, and I'm just kind of ignore some of this stuff here, but basically looking at figure G are images of these cells, or representative images, I guess, from micrographs. Morphology of IBA positive cells, which are microglial cells. So they do like microscopy, and you could see the morphology is quite different. So one thing is that young ones have these long processes and kind of smaller bodies, or kind of soma. It's kind of hard to see. And here the old, it's a little bit bigger and smaller processes. And then that indicates that, I guess they're engulfing stuff. I guess they're kind of active, and I'm not sure why the processes would be shorter, but that's kind of a morphological kind of indication of their activity. Go back to the chat here. Bone marrow composition, Luca states that bone marrow composition changes with age as the relative frequency and nature of active hematopoietic progenitors. This in turn affects the capability of young versus old. I'm going to stop sharing here while I read this. This in turn affects the capability of young versus old bone marrow to fast reconstitute hematopoiesis, particularly along the early time window to study. The incomplete or slower hematopoietic reconstitution might substantially impact the behavior of the mice. In simple words, the mice that reconstitute faster would feel better faster than the mice reconstitute. Again, this is due to narrow and very early. They check for the progenitor composition of old versus young bone marrow used for transplantation. I don't think so. I don't recall reading it in the paper. It could be buried in the materials and methods, but that could account for, the huge error bars. So that's, that's a good kind of tip for the authors here is to find out why you have these really wide error bars, because, these are genetically similar mice. They're pretty far inbred. So, what accounts for this broad kind of error spectrum? Is it just kind of, error in the amount of progenitor cells that you seed the mice with, right, initially? Or does it have something to do a little bit, deeper with regards to the speed of reconstitution that Luca Biasco mentioned? I'm not sure, so what's good is despite that, they still were able to kind of see some significant differences. And perhaps, and this is kind of a general observation, if we can kind of read deeper into these papers and try to track down the sources of error, then that could really help out biomedical research quite a bit, because, this variability between individual and individual, the more complex an organism gets, certainly metazoans, is a problem. And, if we see this kind of error in congenic strains of mice, then, it's like, boy, the error is all over the place with one human versus another, right? So, so tracking that down is key. So let's get back to it. Okay. So let's see. I'm gonna sort of, well, I could go through everything here. So let me just pick something here. So they're looking at staining. So they're doing staining with these markers called LAMP1 and CDC68, which are indicative of activation of lysosomes. So, lysosomes are basically where things get broken down. And if things are getting broken down, then obviously, evidently, or, perhaps the cells are in the midst of phagocytosing stuff or doing something active, right? So if we go to cells that are IBA microglial cells, microglial cells, are they LAMP positive? The numbers go down with young, but they also go down with, just going to M with your old mice with young bone marrow, and also CD68, another marker, relative expression, and also specific for IBA microglial cells. And this soma area and process complexity and length, this has to do with these figures here, which is, again, morphological changes that basically underlie microglial cells that are active, right? So, not actively scavenging stuff, actively scavenging stuff. So, okay, microglial are active. In older mice, that seems to get reversed, and cognition seems to get better. And that kind of leads to where they sort of really zoom in on one molecule in particular, based on other studies that have implicated it, and that's circulating CCL, that chemokine that I mentioned, CCL11 levels, and they look at that and they say that they're reduced in young bone marrow recipients. And also CCL11 injection into young mice mimic hippocampal aging. So they kind of recapitulate some of the stuff they showed before. They don't do any of the, I don't know if they did any of the memory tests. They basically looked at, they looked at the synaptic plasticity, they looked at the dendritic spines. So here we go, CCL11. The one thing, okay, so I'm going to point, I kind of did a quick literature search. very, very perfunctory. So maybe this study has been done. So it's good that you see higher levels of CCL11 in old mice and lower CCL11 levels in young mice, and then you see this trend recapitulated when you do this bone marrow transplantation, young to old. They didn't see the same thing with this other factor called beta-2 microglobulin, which is it's actually a subunit of MHC class 1. And I haven't been able to find much about it. It does tend to form kind of plaques not related to the brain, but in joints due to kind of other conditions. So it does form this sort of, I don't want to call them, they're not amyloid tangles, but they are sort of plaques. And this was implicated also in memory, but in this case, for the, as a result of this transplantation, whatever benefits of transplantation occurred, it had nothing to do with lowering the levels of this beta-2 microglobulin. So they looked at CCL11, which it seemed to do. And then, of course, PBS, CCL11 here, PBS, those are injections. So they're looking at young mice that have been injected with CCL11. And what they're showing here is that, they're saying that this, again, a lot of error, but statistically significant, that the number of spines goes down. Basically, the activity of the morphology of these neurons, and resembles older neurons, and the activity of glial cells goes up, right? So these cells that have CD68, which is indicative of lysosomal activity, goes up, LAMP1 goes up. Obviously the $1 million question here is, well, what happens if you take CCL, I'm going to stop sharing this. What happens if you take CCL11 out of old mice, right? So I don't know if anybody's, obviously there's two ways you can do that. You can inject antibodies against CCL11 into old mice. You could also perhaps run the circulatory system from an old mouse and somehow run it through some kind, I'm just spitballing here, through some kind of column or some kind of dialysis membrane that has antibodies for CCL11 or some ligand for CCL11 and clear it out of the blood, right? So is CCL11 something that, that would add much more strength to this hypothesis here, right? So adding in CCL11 to young mice does bad things. CCL11 levels that are lower when you do this, amongst other things, lower in old mice through the bone marrow transplantation seems to confer benefits. But is it specifically CCL11? And could you design then an intervention where you just target CCL11 rather than doing whole blood plasma transfusions? So Lucas is not talking about the age of the mice, talking about the time after transplant. Can they survive for 12 months after transplant? Yeah, so if we go back to, let's see, share my screen. Let's go back to the graph. Back. Okay. So yeah, so they do behavioral testing 6 months rather than 12 months. So I guess then the question is, could they do this now 30 months, right? A 30-month-old mouse. I guess, they do behavioral testing 6 months post-transplantation. And they're saying that the reconstitution is in 3 months, and you're saying that it should take longer, and maybe they'll get better results. I don't know, perhaps. I'm gonna, oops. Well, we can Google that later. Let's see, where's the chat? I'm gonna stop sharing. Oops, stop sharing here. Despite being relatively old, I believe so. Yeah, I don't know. I don't know what's the maximal age of off the top of my head, C57 black 6 mice. How to do physiological testing. And I have Keith, for some bizarre reason, calling me in the middle of the journal club. Keith is the president of livestreamed.io, so hopefully his apartment isn't on fire. But that's a good question. So I'm going to go back. Oh, God. Keith. Steve, if you could let Keith know that he can just chat during the journal club if he wants to ask us questions instead of phoning in.
I'm already on it. I've just said, dude, stop calling us.
Yeah. Okay.
I don't know. It's these fans. They're such a pest, aren't they? You know.
No, the fans are great. It's
No,
cool.
you said he's your biggest fan. Keith said.
He's still calling. Let me. Well, we're pretty much close to the end here. Let me just scroll down here. So what I wanted to kind of mention is, yeah, I'm really kind of excited that we could do a follow-up study with, getting rid of CCL 11. That to me is kind of the most direct follow-up study. Do we have elevated levels of a similar, chemokine in human cells? Is that what was the chemokine that was implicated in the paper with Amy Wagers, going way back where there's some controversy? That was a different
IGF-11.
Yeah, it was an 11 in there, but maybe I don't know if it's the homologue of it. I'm not sure.
It's IGF-11. I'm going to kill Keith. Keith won't be making it to longevity escape velocity if he keeps pestering me.
Okay, so going back to the paper here, what they do mention is a clinical trial. Share my screen here. Share this one here. So if we go to, if anybody's interested in looking up clinical trials, there it is, at least in the United States, clinicaltrials.gov, NIH, and then you can type in a condition or disease. So let's put in Alzheimer's, and they mentioned that there's a acronym called PLASMA trial. Those studies, PLAMA, I just misspelled that. Plasma study, there we go. And this is a study that just finished. There we go. That was mentioned in this paper. Basically, it was a 4-week study, very short, where they did transfusions for people who had Alzheimer's. And if you're wondering, did this actually work? Well, they weren't really testing for that. They wanted to see if this was going to be, if the individuals, this was, finished early this year, the individuals were able to kind of, tolerate this, because these individuals had mild to moderate Alzheimer's disease. Can they keep this study going? So it's a double-blinded design with a placebo control. So 1 unit from male donors, placebo infused once weekly for 4 weeks. So bottom line is the trial was safe. They didn't really look at any, there was no, in that very short window, there was no really, I think they may have done a mini-cog test. They didn't really see much improvements. They did note that the caregivers noted that that I guess maybe the energy levels of the participants were better, so that there was a subjective improvement. Bottom line is that we're gonna, everything points to more studies along this, along this kind of thread of not only plasma transfusions, but looking at the factors that are responsible in these, for these effects, both negative and positive, in transfusions. The CCL11, the fact that it's a decrease of a factor is pretty cool because, drugs that knock things out, I mean, that's what pharma's pretty good at, is coming out with things that knock out the function of something. So if something is activating, something else that's activating inflammation, then we should be able to knock it out. And of course, there's much more follow-up questions that we can have. Are any of these factors, any of these chemical compounds like Physidin or curcumin or a million other compounds that are known to have all these protective anti-inflammatory effects, are they having specific effects on these factors that are found upregulated in older blood plasma, right? So there's a lot of follow-up experiments that could be done here. So I think it's a very good foray into this field from this paper. And it's, and again, we're right at the threshold and of doing clinical trials. Actually, people are already doing clinical trials, but the stage is now set for doing phase 2 trials, etc. With more participants, now that the safety of a lot of these, compounds and treatments have been validated. So the trial that just finished, I just mentioned now, the plasma trial with mild to moderate Alzheimer's patients, which is really kind of a phase 1 trial. Also, the trial that was done with senolytics, very preliminary, but in human patients with, dasatinib and quercetin, with, Kirkland and group at the Mayo Clinic. So we're going to be seeing a lot more trials coming up in the next few years with larger cohorts of patients. That's something that we will bring to you once those results come in. So, yeah, I have nothing else to really say at the moment.
Wow, really? Well, there you go. He's blown away by it all. But there's been a few bone marrow transplant papers recently. There was another one that we published a few days ago, an article about it where they actually look at mesenchymal stromal cells in the bone marrow, Oliver, and it seems that these particular cells work in unison with the actual blood producing cells, the HSPCs, which sounds like a bank, but it's not. And
that's HBC, I believe.
HSBC, yeah. Yeah. But yeah, they apparently they work in unison, and they found that senescent cells play a role, that the mesenchymal stromal cells start to become more and more senescent-like, not necessarily directly senescent, but it's a more gradual state. It's not so analog, and the more they drift towards senescence, the more they secrete the SASP, and it interferes with the production of fresh blood cells and such like. And it was quite interesting, and it messes the whole immune system, and it's really been directly correlated with immunosenescence. So there's definitely a lot to bone marrow, and I have heard some researchers like Stoltzin, for example, they've always felt that if we could actually rejuvenate the bone marrow comprehensively, there's a great potential for resetting epigenetic age of, blood cells, and by proxy, that would also mean immune cells as well. So I think there's definitely very interesting, and also to touch upon what you said earlier about filtering. Of course, Irina Comboy and Michael Comboy have sort of been moving that direction for a long time, for well over 10 years, which also ties in with Amy Wager's. It was GDF11, by the way, not IGF. All these numbers, that it becomes a blur after a while, doesn't it? Yeah. But it's GDF11. But all these factors, they're starting to really sort of work out what does what in the blood, and it's not beyond the realms of plausibility to filter these, and that's certainly what Irina Comboy and Michael are moving towards. And
right.
And
even a low-hanging fruit, even before getting into filtration. I mean, it's a good likelihood that some of the a hypothesis I'll throw out there is some of these anti-inflammatories might be binding at some degree of specificity, glomming on to these chemokines in the blood and basically preventing them from docking to their cognate GPCR on the glial cells. And that's something that could be looked into. That's a simple, if I have had some money and a research assistant or 2 or 3, it's some assays that we can put together and see if that's what's happening.
Money, but surely scientists are extremely well paid and well funded, are they not?
Yeah, well, I mean, I've got this drawer full of stacks of $100 bills right here, but that's for my sundry hobbies. It doesn't really go into.
That's for lighting your cigars, your Cuban cigars from, isn't it?
Yeah, that's the personal fund. The big stack of the $1 million I got in there. That's
exactly a note for everybody at home there. If you are going to smoke fine Havana cigars, do make sure you do light them with $100 bill.
And if
no
you break
less.
And if you break into the Cambar lab and you look into that drawer, I'm sorry to say what you'll find is stacks of old scientific papers with scribbles on them.
And maybe some moths. Some moths. Let me out. Yeah, but joking aside, yeah, funding is probably one of the greatest bottlenecks to progression in aging research. We ask a lot of researchers what's the biggest bottleneck, and invariably it comes down to funding rather than the actual technical challenges of solving aging. It's the funding issue that most seem to sort of strike upon, don't they? So there you go. That speaks volumes. People are saying, well, we could probably do something about it, but we can't even keep the lights on. So there you go. There's a thought for the day.
Yes, indeed.
Oh, and yes, and a message for you, Oliver. Keith says, could you please phone him when you finish?
I will.
Oi. So anyway, that was great. It's very interesting. I've been following this, this particular area for quite a while, and it's definitely exciting. And I think we're not far away from potential solutions in the next decade or so. And that's about it, really. And we are in Berlin, are we not, next week?
Date is approaching.
Yeah, it's been a long time coming, but we are away, which is why we've been a little bit early this month with the Journal Club, because we are all going to be over in Berlin for the Undoing Aging Conference, which starts on the 28th of March in Berlin and is hosted by the Forever Healthy Foundation and, of course, SENS Research Foundation. And we are very thankful to them for inviting us back this year. And we will be there in force. We're going to be doing lots of interviews and all kinds of things. So watch our website and look out for the articles in the following week or so, where we will reveal all, or certainly tell you what the researchers were willing to tell us. Although we won't be telling you about any of the top secret experiments, right, Oliver, like the Man Bear Pig project or anything like that. We can't talk about that.
Wait, the which project?
The Man Bear Pig project.
Oh, the Man Bear Pig, yeah.
Yeah, Man Bear Pig. So we'll be under an NDA for that, so we can't really say much more than that. But we will be bringing you what we can. So stay tuned. We'll be back next month, I suppose. I mean, we've made a bit of a habit of it, haven't we?
Yep.
So we'll let you know through the usual channels what the topic is. As always, if you do have a suggestion for a paper, drop us a line and we'll have a look and see if we can convince Oliver about it.
Okay.
And that's great. So once again, I'd like to thank the Lifespan Heroes for their continued support. And if you'd like to check that out, if you visit www.lifespan/heroes, Hero, if you want to learn about how to support us. And that's great. Thanks for joining us. And we'll see you in about 5 weeks' time. I think so, about 5 weeks. Thanks for joining us, folks.
Okay, everyone, take care, stay healthy, and see you soon.